Hello, everyone. I'm really happy to be part of this Korea event for the first time. And I wish I were able actually to come to Korea and visit your beautiful country, and hopefully next time. Also very happy to be part of this workshop that we organize at CILcare with the support of Jay, Korea, and also the different speakers from today. So thanks a lot to our speakers, Helen, as well as Greg and Sang Yong. Really appreciate that you were able to be part of this workshop, and hopefully we can also do more work together in the future. This workshop today is really an important one because it covers a huge unmet medical need. So the two indications that we will talk about today are the noise-induced hearing loss and age-related hearing loss. And we'll try to cover different aspects that are related to these two very important aspects or indications of ear disorders. The first one is age-related hearing loss. This is a major and increasingly prevalent hearing issue or health issue. And also it's one of the leading causes of chronic disability in elder populations. It usually occurs in most mammals with different variations in age of onset, the rate of decline, as well as the magnitude of degeneration in the central nervous system, as well as the inner ear. Also, noise is a very common cause of hearing damage, which can lead to synaptopathy and ultimately neuronal degeneration over time. So this workshop aims to discuss the latest research about aging and effect of noise-induced hearing loss. So we'll talk about the physiopathology of hearing loss and the impact of noise exposure to mainly gain some insights on possible treatments for these important indications. So the title of the first talk of today's workshop is mainly about the noise-induced hearing loss and aging, where we will cover subjects related to physiopathology and some of the strategies for preclinical development. Before I speak about these two important indications, I would like to only give some few words about CILcare. CILcare is a CRO, and it's the leading R&D partner for external innovation in hearing disorders. So if you are a pharmaceutical company, a nutraceutical company, a biotech, a medtech, or a vet company, then you are interested into going into the hearing space, you would need some partner that has expertise in this field, and CILcare claims to be that specific partner for you. CILcare has these are some facts about CILcare. CILcare has two state-of-the-art auditory laboratories in France, where we also have the headquarters, as well as the US in the Boston area. We have this alliance with an American company called CBSet back since 2017, so 4 years ago. We also have a business office in Paris, where I myself am based, and we also opened a new subsidiary in Denmark back in October 2020. Since our inception back in 2014, so about 7 years ago, we have worked with more than 70 partners from different continents like Europe, the US, and of course Asia, where we perform more than 130 auditory studies. So in the coming few slides, I will be speaking about the epidemiology, the etiology, the physiology, or the physiopathology of hearing loss. You can see here that by 2050, and this is according to the World Health Organization, that 1 out of 4 people will have hearing loss. And you can see also that the West Pacific region, where Korea is concerned, is also one of the most impacted regions. So that means that hearing loss is a global public health concern with a dramatic increase in prevalence, and that's why it is quite alarming. And you can see also that the highest rise projected in the next decades. And this is again, as I said, that Asia and also Korea will be very much concerned. If we look at aging, we'll see that aging is a major risk factor for hearing loss. Even though this is true, it doesn't have to be unavoidable. It can actually be unavoidable, and that's what we're going to try to speak about today. And if we look at the different risk factors of hearing loss, we'll see that the main ones include aging, excessive noise exposure, the intake of ototoxic drugs. Right now we have almost more than 200 ototoxic drugs in the market, which is really alarming. And of course, there is the chronic ear infection that is usually established or associated with children. So that means otitis media, which is usually untreated in developing countries. And of course, there is aging, which is known to be the major cause of hearing loss. It results of genetic influence. So far, there are more than 250 genes that are associated with hearing loss, as well as health conditions. So comorbidities, here we speak about examples like diabetes, hypertension. So these diseases makes patients more susceptible to developing hearing loss. And last but not least is the lifestyle. An example of that is excessive and prolonged loud noise. It can also lead to an irreversible hearing loss. So our teenagers really need to be careful, and we need to tell them about that. So you see that in this slide that age-related hearing loss is really one of the most common conditions that affects seniors. And it's part of the natural aging process. So it's really part of the senescence. And usually it cures gradually and affects both ears. And most of the time is also associated with cognitive decline, like what we see in dementia. Today there is 1 out of 3 people that are over than 65 years old suffers from disabling hearing loss. And these numbers, if we even go down in the population to 45, you will see that 1 out of 4 also suffers from hearing disorders. So if these are unaddressed, if these disorders are unaddressed, it might lead to communication difficulties. It leads to isolation, it leads to depression, reduced quality of life, as well as loss of independence and mobility, specifically for our seniors. So hearing loss at high frequency gradually develop with aging. This is quite characteristic of the age-related hearing loss, and as always also obvious, it coincides with difficulties on speech perception or speech recognition. So age-related hearing loss can be defined as a progressive, bilateral, symmetrical hearing loss due to age-related degeneration, and it can be considered as a multifactorial complex disorder with both environmental as well as genetic factors that both contribute to the etiology of the disease. The free radical theory of aging that were proposed by Harman more than 50 years ago has actually obtained consensus by several lines of evidence, and the mitochondrion is reported to play a key role here in aging as main source of reactive oxygen species, what we call ROS. If we look at how it's being diagnosed, age-related hearing loss is usually diagnosed by looking at the medical history of the patient, by doing audiometry, the very common audiometry, and also tests of speech perception. And if we look at the different existing solutions today, we see, of course, hearing aids. This is mainly for people with mild hearing loss. But then if the hearing loss is severe, we need to switch to cochlear implant surgeries. So these are quite expensive also surgeries. And these two solutions, of course, they are great. They help a lot of people, but they're not sufficient. So for tomorrow, we need to find other therapies. We need to find other solutions. And the good news that with that we see a lot of investigational drug candidates in the pipeline of lots of small, mid, and also large pharma and biotech companies. One of the things that actually opens, the door for hopes to find therapies is the ability to now understand the ear anatomy, and then ability to do local administration of drugs, which opens the door for drug repurposing strategies. And if you look here at this graph, you will see that some of the pathways that are involved in the pathogenesis of hearing loss, they are actually also common to other diseases. And because these pathways are also related to many other diseases, there are drugs that have been shelved that now we can reassess for efficacy in hearing indications. And the reason for that is that because now they can be administered locally, which will of course reduce their potential side effects like systemic exposure. And of course this is linked to the drug repurposing strategy, which is now becoming more and more spread. And because this cochlea that you can see in the left side, which receives the sound, is really difficult to target, and the signal sent by its hair cells must be understood by the brain, it requires a very special expertise, the type of expertise that people at CILcare has. And if you look at the age-related hearing loss, you will see that it's also attributed to different changes in the peripheral auditory system. Usually there are three different types of changes. It could be sensory changes, metabolic or neuronal changes in the peripheral auditory system, which usually in the end affects the hearing ability. The first one, the sensory changes, are usually the degenerations of the outer hair cells, as well as the inner hair cells that usually starts at the base of the cochlea and over time progresses to the apex. And this is really leads to age-related hearing loss, and you can see that evidence in the high frequency threshold rises in the audiogram. In the metabolic age-related hearing loss, with atrophy of the stereo vascularis, you can see that the audiogram is characterized by a flat hearing curve. And when there is damage, of course, to the spiral ganglion neurons, this is usually classified as neuronal or age-related hearing loss, and audiogram is unaffected, but there's always a poor speech perception, especially in noisy environments. Let's dig into the molecular pathways that are involved into the age-related hearing loss. There is an increasing evidence that links oxidative stress to age-related hearing loss. So oxidative stress can cause irreversible DNA damage, and there's also the incidence of mitochondrial DNA mutations, which increases with age and frequently also is observed with age-related hearing loss. There's also the accumulation of the senescence cells that is involved in the physiopathology of this disease. And finally, several pathways that includes oxidative stress, triggers inflammation, and increases markers like IL-1 and TNF levels. All of this highlights and supports the development of pharmacological therapies with antioxidants, mitochondrial metabolic regulators, as well as apoptotic or apoptosis inhibitors, and finally neutrotrophins that protects the cochlear hair cell or protects the cochlear cells against age-related hearing loss. What if we look at both age-related hearing loss and noise-induced hearing loss? If we do that, we'll see that actually noise exposure early in the life makes the inner ear more vulnerable to aging. So noise exposure is well known to cause permanent or transient hearing loss, and underlying molecular mechanisms of noise-induced hearing loss includes oxidative stress as well as inflammation, specifically or exactly as we just described in the case of aging. And there's also in this case the glutamate excitotoxicity as well as calcium imbalance, which also lead to activation of cell death pathways in sensory hair cells. And actually lots of recent research demonstrate that noise exposure in early life could worsen the age-related hearing loss back in the days or later in the days by triggering progressive neuronal loss. So now I'm going to switch gears a little bit and talk about the preclinical development strategies for both these two indications. This comes from our own expertise at CILcare, because that's exactly what we do, and you can see that in this graph. There is also one thing that I like to repeat, is that when we are thinking about preclinical development, we also need to think clinical development. So we really need to have in mind what kind of clinical indication that we would like to target before we start the whole drug discovery in the preclinical phases. So usually this should include thinking about the route of administration that also you are considering in the clinical phase, as well as the endpoints or the outcome measures that you are interested in the clinical phase. So usually when you are done with all the pre-formulation work and you are sure that your drug is able to reach the inner ear through all the PK studies, then you need to select the best efficacy model for your indication, which also includes defining the drug regimen, the route of administration. So this could be systemic route or local route. And then if you go on to choose the local route, which one? Is it transtympanically or into the round window niche, or maybe even via the intracochlear injection? And of course, you need to look at the most translational, but also maybe some complementary readouts. This is usually what is happening in the efficacy study or a proof of concept study that is also followed by an autotox study or general GLP autotox study, which we will not be covering today in this talk. So let's dig in one of the models to look at the age-related hearing loss in animals. And this model is the SAMP8 mice model that we have at CILcare. And this model can be used to assess the effect of a compound on the auditory as well as the cognitive functions. And this SAMP8 mice strain usually displays a remarkable age-related deterioration. And you can see that we have also a diverse set of functional as well as behavioral readouts that I will be explaining in the next few slides. Then why did we choose SAMP8 mouse? Lots of different reasons. The SAMP8 mice strain is an excellent model for aging research, and it displays a premature hearing loss and cochlear degeneration, so the same as happens in humans. And this is a senescence accelerated mouse. They are mutants, but they are not genetically modified mice. So the main characteristic of this mice is that they have a very high oxidative status, and they represent a model for accelerated age-dependent disorder, including hearing and cognitive impairment. The SAMP8 mice exhibits a shortened lifespan. As you can see in the graph here, it's about 11 months, and usually hearing loss is observed from the age of 5 weeks only. If you look at the right side, you will see the graph that represents humans. You will see that the aging process accelerates from 50 years old onwards, and you can see that in SAMP8 mice, this process really starts early, almost from the time of birth. That's why it's really a good model. So in this slide and the next few slides, I would like to give an example of a study that we did conduct at CILcare back in 2017, and you can see also that the results are published in 2018. And this study included the NAC, or N-acetylcysteine, which is a known antioxidant, and this was the study design, what is usually the default study design of this model. So it usually includes a minimum of 2 groups if you're going to only look at one dose, one concentration, and the number of animals per group is usually 15 animals. And usually 2 groups are vehicle group and a treated group or compound group. And the study here, it runs from 10 to 14 weeks. And at T0, all the animals, after they are acclimated, of course, we perform auditory baseline measures of both ABR and DPOE. And we usually measure these auditory parameters every 2 weeks. And in parallel to that, we can also assess the cognitive functions with different behavioral tests at baseline and, of course, at week 7 and at the end of the study. And at the end of the in-life phase of this study, we can also sample the cochlea to count the hair cells. And for the different readouts we have, you can see in the bottom here of this slide, we have 4 readouts to assess hearing functions. The 2 first ones are complementary functional measures. These are ABR and DPOE, and they are non-invasive. They are translation readouts, and they are used for screening newborns. The first one is the ABR. These are they are measured by electric potentials and that are recorded from scalp electrodes following a sound, and usually they represent the activity of the inner hair cells as well as auditory neurons. The second readout is the DPOE, or distortion product autoacoustic emissions, and these are acoustic responses produced by the cochlea. So this usually reflects the functionality of the outer hair cells. So now we have 2. So that's why we say complementary, the outer hair cells as well as the inner hair cells by 2 different measures. And then we have the cochleograms and the scanning, so the scanning electron microscopy, and these are histological analyses that we can use to look at the hair cells, quantify them, and also look at the morphology of these hair cells and determine if there is hair cell loss at specific frequencies or not. In the end, we also can do correlations between these different types of readouts to confirm the results. So let's look at some of the results of this study that, as I said, it was published back in 2018. And let's start with the ABR thresholds. So you can see in this model that that we have tested the effect of N-acetylcysteine, which is, as I said, it's a well-known antioxidant. This snack or antioxidant was diluted into the drinking water. So it's like, you can say it's an oral administration. And then the mice were treated for 10 weeks, which is quite the minimum if you would like to run this study. And we measured ABR thresholds and DPOE amplitudes at different time points. The NAG group is represented here in red, and the vehicle group, which is just drinking water, is also represented in black. And at day 30, so the first graph on the left, the first curve on the left, you can see that both parameters were similar to in both groups, which is expected, and we observed then a progressive increase of ABR threshold of the vehicle group, which demonstrates the onset of the hearing loss. However, the NAG significantly delayed the progression of ABR threshold shift in mice from 45, so you can see that 45, 60 days, as well as 75. These 3 graphs that also are highlighted with these orange circles. Sorry, next slide. Here you see that another example of the results of this study, and that's the effect of the antioxidant on hair cells. And in this one, using the scanning electron microscopy, and you can see that it actually the NAG has been able to reduce, or has been able to deteriorate or prevent. Sorry, it has been able to prevent hair cell loss, so either inner hair cells or outer hair cells, without affecting the morphology of these hair cells, which is really good results. And now actually I will switch gears from the age to the noise-induced hearing loss models. And at CILcare we have actually 3 different types of models to assess noise-induced hearing loss in 3 different species. So these are mice, rats, as well as guinea pig. And the first one is the TTS, or transitory threshold shift. This is for assessment of temporary hearing loss as well as synaptopathy or hidden hearing loss, and this is we do at in SAMP8 mice, in CBA mice. And the two other models are these are PTS models, or permanent threshold shift models. These are for the assessment of permanent hearing loss in both guinea pig. In this case, we use Hartley guinea pig, as well as in rat, and we use Western rats. You can see that these models have different trauma characteristics, but they are all bilateral, and the readouts that we use for all the three models are the same. So all functional measures like ABR, DPOE, and in this case we also can use measure wave 1 analysis, as well as the histology parameters like cochleograms to do hair cell counting, and we also can do Braven synapses and CGN or spiral ganglion neurons counting. So let's look at one of these three models for noise-induced hearing loss, a little bit of more with more detail. And I chose here the permanent threshold shift model, PTS model in guinea pig. This is an example of a study that is a scheme of our study design that we usually run. You can see here that it's a permanent threshold shift model, and the duration of the study is about 21 days. It can be longer or shorter, but it's usually can run longer, but shorter is not recommended. And usually after the acclimation period, we do a baseline measure of hearing functions, that's ABR and DPOE. And acoustic trauma is performed at T0, and at T24 hours also we do another measure of auditory functions, mainly to check the effect of the trauma, as there is some variability due to the individual trauma sensitivity of the different animals. So other measures of ABR and DPOE are also performed until day 21. After that, we also do a sacrifice, and we look at the cochlea and we perform cochleograms or histology. And regarding the compound administration, we can also do all different types, as I mentioned before, either systemic or local administration. And when to do that, when the drug regimen. So generally speaking, the timing of the administration can be just before the trauma for a preventive approach, or just after the trauma to protect the hair cell damage. So here I just put an example of the validation data that we obtained from this model in guinea pig, and you can see here that at baseline, and that is represented here by the brown line, the animals displayed a normal value of ABR thresholds, that is to say below 20 decibels, and also displayed a classical pattern of DPOE amplitudes. This is exactly what we expected. And you see also that 30 minutes after the trauma, there is a significant increase of ABR thresholds at all different frequencies. And the curves at 14 and 21 days confirms the permanent hearing loss with significant increase of ABR and a decrease of DPOE. If we look at more results of this validation study of this guinea pig model of noise-induced hearing loss, you will see that, and this is the cochleogram results, you can see that the acoustic trauma leads to hair cell loss. So this really validates the model, and you will see that on those cochleograms, we can perfectly see the loss of the hair cells in traumatized animals as apparent on the second image, or the image below of this basal fragment of the cochlea, where cells are labeled with anti-myo7A, where you can also see that it corresponds to the graph on the right side here of the slide, as hair cell loss is concentrated around 8 kHz, which is the frequency range that we did target. Before I end my talk of today, I would like to leave you with a few take-home messages of this talk that might be a little bit too much information, but at least you would, if you would go home with these few take-home messages, that would be great. The first one, ear disorders are global unmet medical need, which is really increasing dramatically and needs to be addressed. And then a second take-home message is that age-related hearing loss is one of the most common conditions affecting seniors all over the world, and that noise exposure early in life makes the inner ear more vulnerable to aging. So we need to avoid that, and we need to find solutions. And number 4 is that local administration opens the door to drug repurposing strategies, which is really great news here. And last but not least, it is important to know that working in the field of hearing requires a very specific expertise, understanding, the ear, and then of course it requires robust, validated, and translational models. This is really evident if you would like to go into the hearing field. So thank you a lot for your attention and for your time. Please, if you have questions, don't hesitate to reach out to me via my email that you can see here, wahid.awad@CILcare.com, as well as at the platform also of BioKorea. So don't hesitate to reach out to me, and I leave you to enjoy the rest of the talks of this workshop. So I revert back to you, Jay, and thanks a lot again. Bye-bye.